Bueno, y finalmente estamos por concluir. Llegamos a la plática final del ponente de H Pincus. Ahora la que se va a cambiar el chip soy yo para presentarlo en su idioma como se lo merece y darle la bienvenida. Before we begin with this last presentation, I would like to thank Dr. Pincus for coming here today. We know that you come straight to the airport, so we appreciate your effort. And I'm going to read some of experience of Dr. Pincus in this field of microbiology. H Pincus is a research fellow of microbial identification in R&D microbiology at bioMérieux and St. Louis facility. He has a Bachelor of Science in Microbiology and Public Health from Michigan State University and a Master in Science in Microbiology from Long Island University. He has 39 years of microbiology experience with a specific focus in identification, product research, and development. He has over 30 publications in peer-reviewed scientific journals and textbooks, including 6 book chapters. His specialty is yeast identification and taxonomy, and he is an editor for medical mycology and serves on the editorial board of the journal Clinical Microbiology. There is also a specialist microbiologist with National Registry of Certified Microbiologists on the American Society of Clinical Pathology, and he is a subcommittee member and advisor for Clinical Laboratory Standards Institute. It's another call with you. Thank you very much, and without further ado, here is Dr.
Thank you for inviting me. I just move this up a little bit. We got it. Can you hear me in the back? Yeah? Okay, good. So today I want to talk to you about the MS, the MALDI-TOF MS system. This is what I'll cover. Some of the slides you may have seen in the past presentation. So I'm sorry for for some of the repetition. It's not so much. I'll talk a little bit about the background, how MALDI-TOF works, how our databases were created, the algorithms, how they work, how you use it in the IVD mode, and then also how you would switch to the other platform, to the RUO mode. And also, I'll show you some performance data from the IVD, from the FDA clinical trials, and also how to build the database on the open side, and what the impact is. So this is an old story, the evolution of microbiology. It was a very slow evolution. It's also kind of a picture of my career. So I started back in the days of Louis Pasteur, with morphology and tube tests. Rapid tests started to be developed in the early 1900s up until the mid-1900s. Spot tests, enzyme tests. This was then the reason that we have manual kits developed maybe in the 1970s, automated kits in the 1980s. Sequencing came along, 16S sequencing came along maybe in the 70s, and it was supposed to replace all of the biochemical kits, but it never really happened because it's kind of labor intensive, it's an expensive method. So today we have MALDI-TOF, which gives the same level of specificity, but much easier to use. So mass spec, it's not new in the chemistry lab. It's been around for a long time, but it's new for microbiology, or relatively new. So it's been around in microbiology for about 10 years, but in chemistry for almost 40 years. MALDI-TOF, that is. And regular mass spec has been around even longer. The difference between regular mass spec that's used in the chemistry lab and MALDI-TOF is that you don't have to volatilize the sample with MALDI-TOF before it gets into the instrument. That happens inside the instrument. Whereas other mass spec methods use volatilization before you introduce the sample into the mass spectrometer. So MALDI-TOF allows for a soft ionization. It allows most of the proteins to stay intact. Whoops, sorry. What happens is we use different types of matrix compounds. The one we use for identification is the CHCA, the cyanohydroxycinnamic acid. If you're looking for smaller molecules like outer membrane molecules, you might use dihydroxybenzoic acid. You might use cinnapic acid for larger molecules in the upper end of the range. So this is a cartoon of how MALDI-TOF works. You put the sample in, a vacuum is pulled in the loading area, and then the laser will fire on the sample. It volatilizes the sample. It's ionized in combination with the matrix. The matrix is a proton donor. And the ions will travel through a high voltage electrical field that accelerates them into the flight tube in a uniform fashion. So they travel then through the flight tube based on their mass and charge. The small molecules are traveling faster and the larger molecules take longer. They hit the detector and then you get a spectrum. And that spectrum can be analyzed by different types of analysis methods. So like I said, MALDI-TOF was described back in the 80s, so more like 30 years ago. There were two main groups, these German guys and the Japanese guys. The Japanese guys were using like a metal matrix compound, and the German guys were using something more similar to what we use today. The Japanese guys probably had better connections because they won the Nobel Prize. And the German guys kind of got left off to the side, but these guys really contributed a lot more to what we do today for identification. So what we're looking for is proteins and polymers in the 2 to 20 kilodalton range. Actually, on the IVD side, we use from 3 to 20 kilodaltons. On the RUO side, we're collecting from 2. Other areas of interest are, things like enzymes and resistance proteins, things like that. Virulence markers are in this higher range. Smaller molecules like outer membrane sugar, peptidoglycan type of things are seen in the lower mass range. So in the mass range for identification, we're looking to detect between 100 and 200 peaks. 20 to 30 of these have been directly related to ribosomal proteins. The others are either post-translational modifications of ribosomal proteins or other housekeeping proteins. It's why we see better specificity with MALDI-TOF than we do with just 16S, for instance, or just 26S for fungi. And these are just some examples of spectral patterns, and the L is for the large subunit ribosomal proteins, and S for small subunit. So these are, you can see that with different species you see quite different patterns. And within a species you have a lot of conservation of the pattern, but sometimes you see some discrete differences. It can be due to the type of source, it can be due to the geography. And it's why we like to collect a lot of data, as many data as we can, have as many isolates as we can to account for this diversity that might exist. Sometimes there's not so much diversity and the type strain is very characteristic. Other times there's more diversity. It's kind of species dependent. So this is an example just to show different resolution power for different types of methods with some different gram-negative types. So, with differential media, chromogenic media, rapid screening tests, you have a quick answer, but you don't have a lot of resolution. You're getting more groupings. With biochemical kits, you have more species-level identification, but the time for result is much longer. It can be hours to days. And the requirement for inoculum is quite high for some organisms. For fastidious organism like a Campylobacter, you may need up to a McFarlane 4, which means you may have to have 2 plates instead of just a colony. Where MALDI is sometimes just using a portion of a colony, like one E. coli colony is way too much. I think I heard earlier, a little bit goes a long way. You don't want to overdo it, because too much, you won't get a good spectrum, good acquisition. And MALDI gives the same resolution as multiple molecular targets. So it's an example of another housekeeping protein that can give better resolution for non-fermented. It's another example of gram-positives. So with gram-positives, you have a lot more choices to make with a biochemical kit, and you can sometimes need greater than a McFarlane 6 if you were running an API strip for coraniforms. And the same thing, it gives the same resolution as 16S plus RPOB for a coraniform or plus SOD-A for a gram-positive coccus. So we have 2 configurations, like you've heard. There's a closed software called IVD, and it has it's been cleared by regulatory agencies. It's IVD CE compliant. It's FDA cleared for almost 200 claims. It can't be modified by the user. The database has today 750 taxa that are comprised in about 700 claims. But the workflow, the software is very easy. And then you have a research platform, which is, the screens are not as friendly, but there's a lot more you can do. It has an open database with many more taxa, but you'll see that it has a lot fewer data. And there's different ways that the algorithms work. So on the closed database, we have what's called an advanced Spectra classifier. And I'll explain how it works in a little while. On the open database, we have either a population analysis, which is called superspectra, or individual spectral comparison, just like a peak-to-peak comparison with the reference spectra, similar to the way Bruker works. And you can generate engrams with unknown isolates and analyze those data that can be collected in a holding area. And then as you characterize them, you can add them to the database. You don't have to characterize every isolate. You have to characterize one in every related cluster. And I'll show you some dendrograms so you'll know what I mean. So this is called the plus. And what the plus did better than what was done separately with the 2 platforms earlier is that now we have the same hardware for the different softwares. So you have the prep station softwares, and you have the acquisition station, the acquisition station here, and the database is here. And I'll show you how the softwares work. First, to tell you how the databases are created. So, wrong button. So this one's a little bit updated from Daniel's slide. So this is the current version of 4.13 SARAMIS. And this is version 2 of the IVD, which is available today. Version 3 is complete, and it's in clinical trials. and it will be released in the coming months. And, outside the U.S., and then after an FDA submission and approval, it will be released in the U.S. So the U.S. will be about a year behind you guys. You're lucky. So, like I said, there's 750 taxa in here. You can see that there's many more isolates and spectra than you see in the research database. The research database has almost 1,500 taxa, but 2/3 of the amount of data, less isolates. And the reason is because there's many more single representatives per species. And when you have a single representative, that's typically the type strain being the basis for the description of the species. And usually the type strain is characteristic. But sometimes it's more of an outlier. So why we like to collect more data on the IVD side, and when we modify the RUO database today, we like to have more data. So more of the species on the IVD side have multiple isolates. The minimum is 2, and the maximum can be hundreds, depending on the species. You know, the common clinical species, we have many. You can see the average number of spectra per species is 40. And here the average number of spectra is 14. It's a slide you saw before. It's a little different. I've changed it a little. Not because I knew. So the population-based approach of the IVD advanced spectra classifier is strictly a population approach. It doesn't have physical spectra like the RUO has. It has a numerical extraction of the spectra. You know, I'll describe how the numbers work, how the calculation works. On the RUO side, the population approach is called superspectrum. And here, not to be stashing groupers, so I just have the RUO reference spectra, which work the same way as a group of biotech. Does it, really matter so much? Sometimes no, sometimes yes. So sometimes you will get a better analysis with the population approach. And on the IVD side, it's a much more robust algorithm, much more complex. So this is the way the advanced spectra classifier works. You have a raw spectrum, we get rid of the noise, and you have a process spectrum with a peak list, and then put into a binning approach. So this binning, it's done with 1,300 mass bins. It's kind of like an API script with 1,300 tests and 751 texts. It would cover the building probably if we laid it out on the floor. But it's huge, and what it has in each bin is the probability or a probability number of whether it's typical to see a peak in that bin or not. And we don't care if there's more than one peak in a given mass bin, it's just whether it's there or not. So what we're doing here in this example is we're evaluating whether this unknown spectrum might be Staph aureus. So in bin 1, the presence of a peak is not typical, or it's not specific for.
Maybe I would do better without the microphone. I'll talk over here. So,
hay que quitar uno. Y el otro bájalo para que no interfiera. Lupita, por favor.
Is it okay now?
Yeah.
So, bin 1 doesn't give any value for Staph aureus interpretation because it's present in other taxa. Bin 2 is highly specific, so it's given a high positive weight. Bin 3, the absence is not so specific, so it's given no weight. Bin 4, the absence is very specific, so it's given a high weight, and so on and so forth. The computer is doing this for 1,300 mass bins. It's a good thing it's a powerful computer, not an Atari. Okay, so then what it does is it evaluates that unknown spectrum against each taxon in the database, and it comes up with a summation score. It's using the specificity numbers, but it's also using the intensity, a logarithmic conversion of the intensity. And there are patents on the data analysis methods. So it constructs a probability curve for each of the taxa in the database and then plots that sum composite against each probability curve to then evaluate the choices. So it evaluates the choices and takes the raw score and the probability against the probability curve for each of the taxa in the database to then decide if it's just one choice or if it's multiple choices up to 4. If it's matching more than 4, then it will give no answer. And if it's matching nothing, it will give no answer. But otherwise, it will give from 1 to 4 choices, and then it delivers the result in myelin. It's an example of a very small section of the bin matrix, the real bin matrix. And this shows you why we're able to differentiate mitis group from pneumonia. And that can't be done on our competitors' product. And I think it's more the way we analyze the data than the number of data that we have. So the workflow is very simple on the IVD side. It allows for multiple prep stations in different locations. So you can have something in a bacteriology lab, you can have a mycology lab. Can't see it, but it's a AFB lab, GB lab, QC lab maybe, or a satellite lab. And what I mean by a satellite lab is it doesn't have to be at that location. It can be in a different town, a different state, even a different country, although it kind of defeats the rapidity of the answer, you know. But if you want to share an instrument amongst different sites, it's possible to do that, as long as the prep target, what I mean by that is that once the sample is put on and the matrix is on and dried, then you can ship that by courier, and it allows for interrogation on one instrument by multiple sites. You've seen this picture before, so the disposable slides are broken into 3 acquisition groups. I think I heard reuse of the groups. I don't know how they're doing that, but when you use that E. coli spot, you could reuse that. Like if I only use 2 spots and I want to fool the instrument by putting a new barcode or just making up a barcode number, I could do that. But that E. coli can't be too old. So yeah, you can do that on the same day for sure. Maybe the next day with a little risk. The longer you wait, the bigger the risk, because the software, the acquisition software is looking for certain peaks to be present in that specific E. coli strain, and it will fail the calibration if it's too old. But a prep target is good for 2 days, so I would expect the E. coli is probably also good for 2 days. An alternative to using unused spots is to use it on the RUO side, because the RUO is not doing the same kind of checking as on the IVD side. There you have to do the checking. You're checking to make sure that the E. coli is giving a good quality identification. On the IVD side, it's checking first for the specific peaks and then later for the identification. What's good about these is you don't have to clean them. If you've ever worked with a stainless steel reusable slide, it's not so much fun. A lot of nasty solvents. You have to be sure that it's scrupulously clean. So in the prep station software on the IVD side, you scan the barcode on the slide. It will show you where you left off. So if you've used an acquisition group, it would be grayed out and it would start on the next group. There's a default configuration screen where you can set wherever the majority of samples is going to be, because there are 2 databases. So you can set it either with the default of bacteria or with fungi, and then you can mix and match along the way if you want to switch as you go. It can be either single or duplicate spots. Normally we don't do duplicates unless it's during training. Or maybe if it's a tube extraction, it's not so difficult to just use another spot. But otherwise, single spots are good enough. Then you would scan the barcode on the plate if you have one, otherwise you can enter the accession number here. And then you would do the deposit of the spots. It tells you where the next one is. If you're doing fungi, it's a light blue to remind you that you need to add formic acid before the matrix. For regular bacteria, we don't add any formic acid, just matrix. And then you validate the data, and then you send the data. There's also a possibility of doing an AST on a Vitec tube. I don't have that in this slide, but that can also be linked with the same data sending to the server. And in the lab, I think you saw a better movie. I don't have a movie, sorry. But the movie was very good. It shows how easy it is. You just take a portion of a colony, or maybe a whole colony, if it's something like an enterococcus or strep, and smear on the target, add the matrix, formic acid first if it's a yeast, and then let it dry and then load it in. On the acquisition software, you would click open. The door opens and there's an adapter that will hold up to 4 slides, so up to 192 samples. It's 4 times 48, but you don't have to fill it up. You can do it however it works with your workflow. You scan the barcode on the slide that you're loading in, and then that will show up on the screen along with the data that came from the prep station. You load it in, you click start, and then after the vacuum is pulled, it will start the acquisition. And after the acquisition is finished, green is good, red is not as good. But usually that's because of not enough sample, too much sample. You can refire on a red one when it's too much. If it's not enough, refiring is not going to help so much. But yeah, if it's a little heavy, then refiring it will read the E. coli again, it will read that spot again, and sometimes you'll be lucky and get a result without having to do any prep. And then you get your results in the result review screen. And this is what they look like. So you have a high quality result, which is a green or a square. Means that your quality control is good, and you have a one choice answer. If you did duplicates, it means you have the same one choice answer in both spots. For a low discrimination or a triangle or yellow, maybe orange to some people, kind of looks yellow to me, means you have good quality control. It's low discrimination if it's a single spot, means there's more than one choice, so up to 4 choices. Or if you did duplicates and you got 2 different answers, where you got maybe one choice on one and multiple choices on the other, that would lead to this kind of result. Or a low quality result is either a failed calibration, a failed acquisition, or no ID. Most of the time it's no ID because it's an organism that's not in the database. MALDI is very specific. It doesn't give the wrong ID very often. So when it doesn't know the spectrum or can't figure it out, it gives no answer. At that time, with a plus, you have the opportunity then to refire in the research software, collect it on the research side, maybe get an answer, maybe not. And if not, you can import it into the database to start to build the database. I'll show you more about that. So we did FDA clinical trials at 5 external clinical sites, large hospitals. These are single spots, no extraction except for yeasts, single firing, and this is with the current version 2 database. So there were aerobic, anaerobic gram-negative gram-positives, yeasts, fastidious gram-negatives. There were almost 5,000 isolates, and we had 96% identified, 4 unidentified, and of the identified, 93% to species, 6 to genus, and 1 raw. The reference method for this, the FDA, they mandated either sequencing or tube testing. So if you've ever done tube testing on 5,000 isolates, you would pick sequencing. Because what do you do with all those data? You have racks and racks of tubes. I used to do tube testing in the old days. I don't do that anymore. Now I use MALDI. So yeah, the reference method was 16S for bacteria, 26S for fungi for the yeast, and sometimes other loci like Recce or Sade or RPOB, depending on the genus. Sometimes we did other biochemical tests, so we had to do an awful lot to get to one choice. And we also had to do analytical studies, so we did limited detection. Limited detection is about 10 to the 5 CFU on the target. Sometimes a little less depending on the organism. We had to do reproducibility, cross-contamination studies to make sure that one spot doesn't talk to the next neighbor. Viability, the matrix kills all vegetative cells. It does not kill spores. It does not kill molds. So we use other methods for that. Won't kill TB. So we have other extraction methods. And we also did mixed cultures to see what happened. Usually when you have a mixed culture, it will pick up the predominant one. Sometimes if it can't tell them apart, then it will give a no answer. Occasionally you might get a little discrimination with both of the mixtures, but it's more typically going to give one answer. So we had a submission of the data and approval the following year. It's a long submission with the first one because it was called a de novo 510K. So now that there is one 510K product for mass spec from MALDI-TOF, the Bruker submission was much faster, and our next submission will be much faster approval. So these are the publications that came from the FDA clinical trials, there were 6. And this is the way the data broke out by groups. So there's gram-positive, coxsackie bacilli, all aerobic ones, facultative, gram-negative enteric, gram-negative non-enteric, yeast, anaerobe, and fastidious. You can see the number correct to species, or the percent correct to species, is really high for most of these. Non-fermenters present more of a problem, and you get more low discrimination. You also get more no IDs because the nature of some of the organisms being so sticky, so crumbly, it's hard to get something on the target. There are ways to work around that, but just with direct prep, this was the result. You can see the performance with yeast is really high for the species level. Same with anaerobes. This is like unheard of with a biochemical method. In version 3 that's coming soon, we have more than 250 new taxa, so that will bring the total in the IBD database to greater than 1,000. We're adding a lot of mycobacteria, 80 molds, veterinary species, about 15 Nocardia, and some other more rare bacteria and yeasts. There are new protocols for molds and mycobacteria, and there's a new acquisition software that works more like the research acquisition to get more high mass peaks that are sometimes helpful for these organism groups. And right now we have a multi-center study in progress. So we have 3 sites for mycobacteria and Nocardia and 3 sites for molds. They're nearly done. So it should be probably finished within the coming months. And we'll use some of those data to release it outside the U.S. The data look very good. Performance is very high. This is the mold prep. So what you do is you take a swab that's a cotton swab that's moistened with sterile deionized water or molecular grade water. You rub like an area of about 1 to 2 centimeters. It's best to kind of get between the conidia and the growing edge, so sort of in the center. You suspend that in ethanol, 70% ethanol, and then you just give it a quick vortex and then centrifuge for 2 minutes. You get rid of the ethanol, either with a pipette or just pour and block. And and then we add 40 microliters of 70% formic acid. It's different than the one you use for yeast. That one is 25%. And then we vortex quickly, just a second or two, and then we add 40 microliters of acetonitrile. And vortex again, and then we spin it again, 2 minutes. And then we're going to use the supernate now for spotting a target. So you spot 1 microliter, you let it dry, and then you add the matrix and let that dry, and then you're good to go. So the whole process for one isolate would take about 10 minutes. It's a little quicker than doing a slide culture or a tape prep. It's a similar procedure. I don't have the one for mycobacteria. It's similar. You take a loophole, and when you suspend it in the ethanol, it's got beads in it, and you put it either on a bead beater or on a vortex attachment and let it mix or really mix hard for 15-20 minutes, and then let it incubate again for at room temp for about 10 minutes, and then go through the same process with smaller volumes, and then at the end it's the same method. So, like I said, when you don't get an answer on the IVD side, the workflow, the normal workflow of the PLUS would be to refire on that spot without doing a new prep, because it's already in the instrument. You don't. All you have to do is define for the research software what to do. So you open up this other software called Target Manager, and in Target Manager, you can skip to the spot of interest, enter the accession number. You can enter other data if you want those other data in your database, like the culture medium, incubation time, temperature, things like that. Or not, it's up to you. The minimum you need is an accession number. And then you would save that plate and then create a project. It's sort of like the adapter on the IVD side. So what you do is you take that plate the target plate that you created, and you drag it into the project. And then once you're ready with either 1, 2, 3, 4 slides, then you would export the data. And then that's where you'll pick them up in the acquisition software. So you would then launch that. And first you have to run the switcher. So there's a little icon on your acquisition software screen. It's called the switcher. You select RUO mode. The Microsoft wheel that we love to see spinning, keeps spinning, and then it goes away, and then you're ready to run the RUO. So you open LaunchPad. It's called MALDI Solutions, Shimatsu MALDI Solutions. And then you, from there, you have a main screen that you access this auto experiment screen. I'm not going through all the steps, but there are a lot of different windows where you can do a lot of different things. But this is just the quick way to get another result. And you pick up the experiment in this experiment genie icon, and then you'll have a pop-up window where you find that experiment. That experiment's going to have a label when it was exported. It's going to show you the label that you created. And then you look for that in the target directory. And then once it's in there, you'll see the data appear for your experiment, and then you would just run the experiment. Then you'll see the spectrum collecting on the screen. If you have good sample prep, the spectrum collection in the RUO is really quick. It could be 15 seconds to get 100 data profiles. Each data profile is an individual spectrum, and then it averages those 100 profiles together to get an average spectrum. And that gets rid of a lot of the noise. And then you have results in the SARAMIS screen. So this is probably on your prep station computer is where you would access SARAMIS. And you would see the results screen where you either have high quality results, the dark green, also good quality, the light green, decent quality, the yellow, a little lower quality, the white. When you have no answer and no matches comparing to each of the reference spectra, then that means you have no ID and it's time to import it into the research database and then have a look at the data to see what you might have. typically you collect multiple data and then look at them over time to see if you have multiples of the same species, and then organize your data in that open database so that you can add new species or new atypical patterns of an existing species. So when you import the data, then you can look at a dendrogram. You just have to locate those data. There's different ways to do that. And then you select the spectra, and there's a taxonomy button where you would then generate, this is a relative taxonomy dendrogram. So it's an example of Micrococcus luteus, where we have one cluster here, and we have another cluster here, which is the type strain. So this is a case where the type strain is not the typical representative of the species. And these are also luteus. And another one to be confirmed, which we find later with sequencing one of them, that it's not luteus, it's some other Micrococcus, and that these were just mislabeled in the freezer, maybe based on an early chemical method or different taxonomy, things like that. Typically the cutoff for bacteria is about 65% relatedness. Some bacteria are lower. Fungi are usually a little bit lower. So it's not a hard rule, but it's used as a guideline. Here's an example of two Aerococcus species. So this is Aerococcus urinae and sanguinicola. We did a study with a hospital in Montreal, in Canada, and they thought they were all urinae. But then when we did sequencing MALDI, we said, no, they're not all urinae, and this is coming in a future version, but today you have to add it to your own database. Similarly, one run on version 4.10 or actually up to 4.13, this will come out no identification. It used to be called Canada Hemalonia, and today it's been described as 4 different species over the years. So they've been broken apart. These cryptic species were described by molecular methods, and we find actually, I did a poster at ISHAM, which is a big mycology meeting that's every 3 years, and this is what I presented, showing that actually when we looked at all of the Hemalonia in our freezer, we had over 40. Half of them were Duobus Hemalonia. It's here. None of them were Orus, interestingly, because Orus is really common in India, in Korea, in other parts of the world, but not seen in the U.S. And can be very invasive organism. They're all very resistant. It's interesting. And MALDI has no trouble telling them apart. You can do peak overlays in LaunchPad, which is nice for publication. So you can plot a few isolates of each species. So these are 3 different isolates of each, and then you can see the peak differences. This is over just a small part of the mass range. It's a nice illustration for publication. So I want to tell you a little bit about how we modify the research database. And it might be a little confusing because there's a lot of steps. Don't worry, you don't have to remember it all. It's just to give you a flavor. And I'll send a PDF of this so that you'll have this, if you like. Because it's got a quick guide to how you would do this. So building superspectra, you like to have at least 2 isolates of a given species. The more the better, because you'll see maybe some diversity within that species. If you don't have a lot of isolates, it's better to use multiple culture conditions, like maybe 2 isolation media, maybe 2 culture ages. So we do that when we only have 1 or 2 isolates of a given species. We'll use 2 media, 2 culture ages, duplicate spectra, so then we have enough to build something with. So you have, in this example, there's 3 streams of species A, and you can see the different peak lists from each. And from that we get what's called a consensus spectrum. So the consensus spectrum is just adding all the peaks together. And you can see the dark ones are present in each of the strains, and the light ones are present in less than all of the strains. And from that, we reduce, my animation's not so good. So we reduce this to this consensus spectrum by taking the ones that are more common in the population. Normally this is enough, and normally this will work to give you identifications of other examples of that species. Sometimes the species within the genus are so similar that you need to use what's called exclusion criteria. And this is a way to create an exclusion criterion. So here we have in the same genus other species and their spectra. And we get a consensus for the genus. And the dark colored ones are common to the whole genus. And we would take that and make that a genus-specific exclusion list. And then we could subtract that from the other one to make it more specific to species A. So like I say, you don't have to do this typically, but sometimes you do. It depends what you're working with. Enterobacteriaceae is a good example of where you would probably have to do it. Because Enterobacteriaceae are so similar by sequencing, they're also similar by MALDI. So this is a kind of a quick guide. So you have this results screen, and they're in this, they're coming from LaunchPad. So they're transferred to this screen by means of what's called an ASCII folder. So the data are put there temporarily, and if they're identified, they're relocated into what's called an identified folder. If they're unidentified, they go to an unidentified folder. And if it's a vast spectrum, they would go to an unknown folder. So it's a way you find your data if you want to find the original files. But you don't need to do that here. Here, what you're going to do then is in that results screen, you want to import all the ones that gave no answer. So you would check that box, that import box. Go back. This one. And then at the bottom, there's an import button. The wheel will spin a little bit, depends how many you're importing. If you're importing 1,000 results, it's a good time for lunch. If you're importing a few, you can probably wait there. Okay, and then once they're imported, they'll go from like a black check mark to a gray check mark. And now they're going to be in this Spectra screen, but you have to find them. Maybe located in either, you can find them either by looking at an unspecified folder, which is down below this. This is the taxonomic tree, but it's compressed. So as we click each plus sign, we see what's underneath. Or you can use a different search criteria. This is by the time of the import, or if they're actually located in, they've been identified and they're located in folders already, you can find them here. So this is that unspecified Spectra folder. It's a good holding place. So all the ones that are unidentified, they go there. And maybe every week or two, you can have a look at the unspecified Spectra folder and generate a dendrogram. And once you see a few representatives that are looking the same, that's when you want to characterize one of them by doing sequencing. And sequencing is really the gold standard here. Running the Bitech 2 is great. We love when you run Bitech 2. And also we like when you run API, but it's not the gold standard. Same with our competitive systems, Biochemical methods are good, but they have a small error rate. And each time you build that into the next product, you're building in errors, so you don't want to do that. So you would search for your imported spectra. You would then, once you know what they are, you would move them to where they belong in the taxonomic tree. So you would create a folder. I won't go through all the steps, but you create a new folder, or perhaps you have isolates that gave no answer, but there are species that's already in there. So you move them to where they belong, and now you're going to compare them to the ones that already are there to see why they're not giving an ID. And maybe you'll mix some of the data together, or maybe you'll just use your new data to create a new superspectrum to get an answer. There's really no limit to how many superspectra you can create for small files, so but typically, if you have a good data set, one or two will do the trick. When you want to generate an endogram, you select the spectra, you click on taxonomy, and then show taxonomy, and then you can analyze the relative taxonomy. The relative taxonomy is typically what we use to define different species from each other. The absolute taxonomy, which is a little further over, is where we identify some of the outliers. Sometimes you'll have just a bad spectrum giving really an aberrant comparison to the others. So you wouldn't want to use that to create your superspectrum. Not necessarily. Sometimes they're a little different, but maybe they add some value. So sometimes you play with it and it may bring some value and maybe not. But typically we start without those outliers at first. So when you've selected the spectra that you're going to use for the superspectrum, there's a filter button here. And this filter button allows you to minimize the data set either by the intensity or the mass range, or using those exclusion lists like I mentioned earlier. Once you've decided what you want to filter, and you may not want to filter anything, if it's a bacterium grown on blood agar, we have blood agar to select as well that will remove hemoglobin-related peak. And then you click on this superspectrum, and here's a graphical view. But here's the view that you would see with all the masses in that consensus superspectrum. You have to locate the data to where they belong in the taxonomy tree, otherwise it won't know what to call the superspectrum once you decide you want to save it. All right, and we select all, and it will show us how many peaks are in the consensus superspectrum. What we do then, go back for a second, is we reduce by the population. So here, it's a little hard to see maybe, but for each peak and the intensity associated with the peak, it shows how many of the spectra that we selected have that peak. So it gives it as a number, like 15 and 18, 17 or 18, something like that. And we select the relative, or the percent of the spectra that, or the spectra that have that peak to reduce by that number. So, and you can't go backwards. So we start a little lower. We start maybe at 70%. Sorry. We start at 70% and then work our way up until we get to a target number of peaks. It may have been maybe 200 or 300 to begin with. And we're trying to get down to maybe 30 to 45. Again, it's a guideline, and rules are made to be broken, so you'll see superspectra that are not always between 30 and 45, sometimes lower, sometimes higher, but it helps to do the math, because the software expects some kind of math. Then we want to compare this draft superspectrum to the database. Then we'll get a statistic of the matches and mismatches. And what we do is we look down the target species to the first non-target species, and we record that number of peaks that's matching for the non-target, and we use that to do the calculations. I won't go into the calculations, but what we're doing is we're going to use these numbers. 1400 is the highest target species weighted sum we want, and 600 is typically the highest we want for non-target. And that's what we use to put in that number code column to give an associated weight with each of the peaks that we have remaining in that superspectrum. So this is more about the math, but we won't dwell on it. And then we have to find the superspectrum in that species folder, and then we select it, and we click activate it, and then it can be used for identification. So it's a multi-step process, but it's not so difficult. It's a little complicated at first, and don't worry if you don't remember, what I said 3 slides ago, because you have to do it over and over again to be comfortable with it. But yeah, and a new super spectrum will give you typically very nice results. If it doesn't, you play a little bit, make another super spectrum, maybe with the outlier, things like that. So, the impact of MALDI-TOF is quite obvious. With IVD, it's a very easy workflow, and it identifies most of the common species you see in a clinical and industry environment. The unidentified ones, or either rare species or atypical isolates, they can be reacquired in the RUO side without doing another sample prep. The RUO lets you build a database with some representative, well-characterized isolates. It's really easy, really rapid. So on the IVD side, it's about 30 minutes for 16 samples from colony to answer. Low disposable cost. The instrument's a little expensive for microbiology. It's pretty cheap for a chemist. But once you get used to that instrument cost, to amortize it over time is very simple because the disposable cost is really low. It's really accurate. Much more accurate than any other method. It's very close to sequencing, but sometimes better than sequencing. It's more specific. So sequencing, sometimes you just be able to tell it's a citrobacter. Sometimes we can tell you more. Same thing with an enterobacter. We can tell different cloacae group apart, where they're really difficult with just 16S. So it consolidates multiple methods. You don't have to select a method anymore. Sometimes you pick the wrong method, you get a bad answer. So no need to decide, much faster, much less code. You can work with a primary plate, which you really can't do with a biochemical method unless it's just for spot test. So that's all I have. So thank you for your attention. Any questions?
Thank you, Doctor, for your presentation, and also Dr. Mexico City. I would like to ask something. This methodology can identify Leptospira and Borrelia?
Not today, no.
Not today.
Sir, not today.
Not today. Okay. And also,
perhaps in the future, the problem is LOD, so you need enough material on the target. If you can cultivate and then clean out the medium, then yes.
Yes, I know. Also, how do you decide to increase the database?
How do you decide?
How do you decide this?
So some organisms were missing.
Yes.
Okay. Some organisms were just not there, important organisms like mycobacteria, molds, or we just had a few and it wasn't as strong as it could have been. Other times we see literature, for an emerging species.
Yes.
We get feedback from our customers saying, why aren't you giving us this answer? And we said, okay, we don't have so many isolates. If you would send us some isolates, it would help. But yeah, all these different ways. And also, when we know of a problem, we also look to fix the problem in a future version.
Okay, thank you.
Hi. So I have 3 questions. So the first one is about the RUO system. So is there a chance that you guys can build a database that users can share? I know that the difference can be different, but you still can get some help somebody else has done, for example, like acquisition of, let's say, Borrelia, and then you can share that with somebody else. Is those bioMérieux share? Is there a chance?
today the RUO can be shared amongst different sites. So there is an export-import function. So if you had Borrelia in a superspectrum, you could share it with.
But in that case, that means that I need to meet somebody that is doing the same thing. My question is that if, for example, the ion torrent, its genomics approach, then they have this website that users can share. So it would be nice if you guys can handle something like that.
I understand. The problem is to be sure that the data are pristine, you know. So when we build the bioMérieux database, either internally or externally, we require a lot of extra stuff in a normal use.
Well, in this case, I know if you want to go like to be approved for the FDA or something like that, then you need to do that. But in this case, it will be something friendly for users, not. So it's just mediated by bioMérieux, but it's not because, of course, you guys can use it, but it would be great if I can meet somebody here, or like let's say in China, and I'm working here, and we're working on Borrelia, like the example we were saying. It would be nice if there's something like that. Otherwise, you need to look around and see who else is working on that. It would be great to do that. That's my question. The other question is, are the updates free?
They are included with your service contract.
That's it. Is there any chance to use this equipment for proteomics or not?
There is, yeah. In the really big RUO manual, the LaunchPad manual, which is 600 pages, it describes how to create your own methods. You have to create your own calibration method as well, because if you're looking in a different mass range, you'll need a calibrant other than the E. coli. And you'll need to check that calibration, but you can add proteins or other chemicals that are defined molecular weight in the range of interest. And I agree with you about the publicly accessible database. It's just for bioMérieux to be doing something like that is really complicated. But there are some other places that are looking at that. Like I know for the fungi, there's a collaboration between the CBS in the Netherlands and also the LMG, the Belgian Culture Collection, and a hospital in Marseille, at least for fungi. I think over time it will come. Also CDC will make available their database. They won't allow you to upload, but they'll make available their database, a data scanning with rare species, and also with high pathogens. It's called Microbial. It's free access. You just have to go to their website and ask for it. They'll send it in there. But yeah, in the best case, it would be great to have something like this.
¿Más? ¿Otra pregunta?
Formular pregunta de la notificación.
¿Cuál? ¿Pregunta?
De la notificación.
Pues ya quedó claro, pero si quieres tengo otra.
Are there a strategy to recognize a particular species in a consortium of 2 or 4 species?
On the RUO side, yes. On the IVD side, not yet, but as an R&D guy, I keep asking for that, because a lot of times MALDI can't tell certain things apart. You can't tell serious group apart. You can't tell, Bacteroidetes from Mesotheria. So I ask for that from the software guys, and I hope someday it will happen. I think we'll have a little more control in the future, but today the MILA connection, since it's linked to so many other platforms, it's difficult for them to give us a priority. So yeah, what happens is the user will manually enter into the LIS instead of having an automatic transfer. But that would be the best case. In most cases, MALDI can tell at the species level. Some even at the subspecies level, but not always.
¿Alguien más? Bueno, thank you very much.
Thank you.
Respecto de una proteína pura, la semipura, pero el problema es que no tienes la base de datos. Y tiene que ser la base de datos para esta parada, porque los espectros demás de los otros no son capaces. Entonces tenemos el modelo y tú ya dices, esto es la medicina. Yo ahora pero desde la medicina tengo
Bueno, no hay ninguna otra pregunta, agradecemos mucho su presencia. Esperamos pronto poderles dar a conocer los lineamientos de uso del equipo para que todos los demoscuchesen los objetivos que ustedes conocieran, cuál es la potencialidad del equipo, cómo lo pueden integrar a sus líneas de investigación para que podamos sacar el provecho que el equipo, o todos los beneficios que el equipo puede dar. Gracias.